Hi everyone, my name is Chatterjee. Thank you to everyone involved in bestowing me with this incredible honor, and especially thank you to Dr. Nigel Fraser himself. I wish I could be there in person to celebrate with you all, but I hope you've enjoyed this short video on my research. Did you know that even teeny tiny microorganisms can communicate with each other? They don't talk like I am, but instead they use chemical signals as their language. One example for why microbes talk to each other is for figuring out their population size. Each microbe sends out a chemical signal, and based on how many of those chemical signals are around, the microbes can count how many cells are in the population. So think of it like a microbial roll call. This allows the microbes to decide whether or not to behave as single cells when they are alone, or as a collective group when they are in a large population. This type of communication is called quorum sensing and has been studied a lot in bacteria, but I study a type of microbe called archaea. For much of scientific history, archaea were thought to be just another type of bacteria, but it turns out archaea are very different from bacteria and are genetically closer related to you and me. In fact, archaea are our ancestors. We don't know much about how archaea communicate, but we do know that they live everywhere. In the soil, in the ocean, and even in our guts. They play crucial roles in the environment and also help with your digestion. So it's important to know how the archaea are talking to each other to get a better idea of how they're influencing these different processes. I work with an archaeal species called Halophorax vulcanii. Halo is the Greek word for salt, and this species is pink and lives in really high salt. They were first isolated from the Dead Sea by a guy named Benjamin Vulcanii, so that's where the vulcanii comes from. You can also find H. vulcanii more commonly around you. If you've ever had pink Himalayan sea salt, you might have eaten some of my friends. I work with this species because they're easy to grow in the lab and they will help us learn about all types of archaea. H. vulcanii exists in two shapes. When there are only a few cells around, they are rod-shaped. But as the population becomes larger, they transition into flatter discs. We think the rod shape is better for swimming around solo, while the disc shape is better for nutrient uptake when packing together and living in a biofilm. I wondered if these archaea are using chemical signals to decide which shape to be. So in the lab, I can trick H. vulcanii into thinking they're in a large population. I grow them in liquid until they are all discs. Then I can take out some of that liquid, which would have a large number of the chemical signals. And when I add this liquid to a fresh set of H. vulcanii cells, they think they are in a large population and turn into discs even when the population size is small. In this way, I found out that these archaea are using the microbial roll call method, or quorum sensing, to communicate their population size. I've also figured out more about the genes involved in how H. vulcanii recognizes and responds to the chemical signals to turn into the disc shape. Additionally, I've recently discovered one of the genes involved in how H. vulcanii produces the chemical signal. And I'm currently working on figuring out what exactly that chemical signal is. I've purified the signal and have provided this material to our collaborators in the chemistry department, who are now working to determine the structure of the molecule. My work showcases the first in-depth study of communication in archaea. And when we understand how archaea are talking to each other, we have a better picture of what they might be saying in different environments, including in your gut after you've had some pink Himalayan sea salt. Thank you.